Hello student, welcome to the next lecture of our fourth week. We have started our discussion about tissue system, in which we have discussed about the epidermal tissue system, which was the outermost layer covering the different plant organs and different plant parts. Now we will move on to the next tissue system, that is the ground tissue system. Now when we talk about ground tissue is all the tissues except the epidermis and the vascular bundle. Okay, so everything except the epidermis and the vascular tissue is considered to be the ground tissue. It is the largest tissue, okay. It is the largest tissue which is found in different plant parts, and it is heterogeneous in nature. Because it contains of different types of plant cell types, and it is not made up of just only one, like it's not made up of only parenchyma or sclerenchyma or collenchyma. All these different plant types contribute to this ground tissue. Now, however, among all different plant types, parenchyma is considered to be the most abundant, is the predominant type. Now, parenchyma, as we all know, that it is the most simple type of tissue. It's the packing tissue, so definitely it will be the most abundant type which is found in ground tissue. And we all know the arrangement of parenchyma. Generally, they will be arranged, and they will have intercellular spaces, but it's still not compulsory that intercellular spaces will be there. But in general, it will be isodiametric in shape. Okay, and they will be generally loosely arranged. Among it, collenchyma will also be present, and generally it will be present in the superficial regions of herbaceous stems and rapidly elongating bodies like leaf stalk and petiole. Okay, so that we have already discussed before, that collenchyma is also a living tissue. However, they have thickened cell wall, and the thickening also vary generally, like on the sides they are more thickened, or however many other various types of thickening could also be there. So it is found in growing tissue and elongating tissue because it provides mechanical support. So the tissues, but yeah, those tissues which are growing and rapidly elongating, they need support, but they also want to maintain their living nature. So in that sense, collenchyma is present. Now, collenchyma may be present in discrete patches. At the ridges, or may be continuous. Okay. Now, along with that, sclerenchyma will also be there. Sclerenchyma fibers will also be there in the ground tissue, and they will also be arranged in discrete patches or in continuous sheet. Now, when we talk about different parts of this ground tissue, generally it is divided into cortex. Okay, you will have cortex, which is the most abundant layer which is found in ground tissue. Now, this cortex will also be divided into hypodermis, then general cortex, then endodermis. That means you will have an outer layer of epidermis. Okay, you will have an outer layer of epidermis, and followed by this epidermis, you will have hypodermis. Okay, just first of all try to make the line diagram only. Okay, so you will have epidermis, and then you will have a layer of hypodermis, followed by cortex, then you will have endodermis. Endodermis will be followed by pericycle. In pericycle, and inside this pericycle you will have vascular bundles, and then in the center you will have pith. So outermost layer will be epidermis, and then you have hypodermis. Okay, then you will have cortex, general cortex. Okay, means actually this is the hypodermis, this is the cortex, and then you will have endodermis, and then pericycle. These will be vascular bundles, and in the center you will have pith. Okay, so these parts you will have, this whole together can be known as cortex. Okay, you can see here it's the general cortex. Now this is it's not that every internal organization will be like this only. This is just a generalized structure which I have drawn. Have a lot of variations you will see in monocot arrangement, in dicot arrangement. Even in dicot also you can have a different variation in different cell types which are present in different layers. Okay, so that all we will discuss later, that how some of the variations are coming out. But these are the different parts of, and along with that you will have medullary rays also. So medullary rays will be in between vascular bundles. They will also be a special type of arrangement of cells that will be found in the central cylinder. Now this is also important to tell you that everything beneath the pericycle, okay, that is also known as stele, means the central cylinder. That is the central cylinder, means the central portion which is involved in conduction. Okay, this whole portion is known as stele. Okay, so this all we have discussed, that it is the epidermis, it is the vascular bundles, it is heterogeneous in nature. Parenchyma is there, collenchyma will also be there, then sclerenchyma fibers. Now in this, see, this is a picture which is showing you some layers of the ground tissue, in which you can see here, this is the microscopic image. Here you can see the epidermis, then this is the cortex. Now this is to show you that collenchyma is present in patches in this particular tissue. Okay, so like if we say that collenchyma is present in patches, so that means, or sclerenchyma is present in as a continuous layer. Now you can have collenchyma arranged in a single or 1 or 2, 3 continuous layer. In generally it is 2, 3 continuous layer. Okay, so they will be thick wall, their corners will be thick. Okay. Okay, then after that you will have loosely arranged parenchyma. So this type of arrangement could also be seen in some organism, in some plant parts, or you can have collenchyma in discrete patches. As you can see here, this is the epidermis. Okay, and you will have collenchyma. Now this type of arrangement you can find in those parts of a plant which can have somewhat wavy margin, like some ridges of the stem. Sometimes you might have observed that some stems have wavy margins. Okay, so in that case you can have this type of arrangement. Okay. Like suppose some stems, it's not that every time it is smooth, or sometimes it might be angular also. So in that cases you can have collenchyma patches. Similarly, sclerenchyma also, which can also be found in as continuous sheet or in patches. Suppose sclerenchyma is there. Okay, so you can have so this is sclerenchyma. Now, sclerenchyma generally you will find in monocot plants, in stem of monocot plant, whereas collenchyma is generally observed in dicot. Generally it is found in dicot. Similarly, sclerenchyma also you can have them as a continuous layer or in patches. So these are basically found in stem. These patches like structure you can find in stem of the dicot and monocot plant. However, when we talk about root, it is more homogeneous. That means it generally contains of mostly the ground tissue contains parenchymatous cell type, although some variations are always there, but in general in root we have observed that parenchyma is the most prominent cell type which is present. So in case of root also you can think it in this way, that suppose it is surrounded by an outer layer and you can have parenchyma. In the ground tissue. Now, along with these parenchyma, collenchyma and sclerenchyma fibers, many other different cell types could also be there. Sclerids, okay, they are also sclerenchyma, but they are not fiber-like. Okay, they have various shapes that we have already discussed, that how different types of sclerids are present. So in ground tissue, you can also have sclerids. They will also provide mechanical support. Okay, along with that, lot of resin means lot of special type of tissues that also we will study. Special type of tissues are there, like some ducts are there, some glands are there. So resin ducts which secrete resinous compounds, sticky substances. Okay, so they are also present in ground tissue. Some oil cavities are there, some latticifers like latex, which we have studied, that latex is a complex molecule which is also secreted by these special type of tissues. So they will also be present in the ground tissue, and many of the things are deposited. Or okay, it's not that the organization of cell is important, but ground tissue serves various function. The most important function is definitely protective layer, and you can say that it forms the main bulk of the plant part. Along with that, it stores lot of substances like tannins, then starch. Okay, they will definitely contain, if they are storing starch, many plastids could be there, then oils and crystals. Okay, they are all common substances which are found in ground tissue. They can also perform photosynthesis, because if ground tissue, like in stem, okay, in herbaceous stem, the parenchyma which is found in the ground tissue may be converted into chlorenchyma, okay, and then this chlorenchyma will have chloroplast, they will have chlorophyll, and they can perform photosynthesis. So in herbaceous tissue also, Suppose this is the epidermis, you can have parenchyma which will be filled with chloroplast. Now coming to the first important part of ground tissue, that is hypodermis. Now hypodermis, you can say it is just present below epidermis, and it is a supporting tissue which is composed of collenchyma or sclerenchyma. Okay, so any layer, okay, this is the layer. If it contains collenchyma or sclerenchyma, we call it as hypodermis. So in monocot also as well as in dicot also, we can have hypodermis. Suppose these are the collenchymatous tissue, we call them as hypodermis. So this is hypodermis, okay, and it definitely provides support because the tissue type which is present is definitely those types which are providing support to the plant. Now after that is the general cortex, okay. These are all loosely arranged. Generally the general cortex is made up of parenchyma, okay, loosely arranged cells. So this is the cortex and mainly made up of parenchyma. Now this layer is highly adaptable, means this particular layer can take different functions by modifying its structure, because parenchyma, as we all know, that it's a very highly flexible type of tissue which can take different forms, like it can be converted into erenchyma or chlorenchyma, and it can store lot of things. So different structural modification could also be there based on different function. So this different structural modification will be definitely present in the cortical layer, like they will change into chlorenchyma, they can have air spaces, and they can be changed into erenchyma. So its thickness can also vary as well as its composition can also vary. It can be developed into erenchyma like in aquatic plants. In aquatic plants, lot of erenchyma is present. As similarly, like in some other, like in rice also, which is present in waterlogged situation, there also you will find lot of erenchyma. Now they contain leucoplasts, that also we have talked about, that if they are stored, these are the plastids, okay, which perform function of storage. So either they are storing starch or oils or proteins. So many things they will be storing. So they will contain leucoplast. Now this cortex will also help in the movement of gases, because generally it is parenchymatous in nature. It has lot of intercellular spaces also. So gases can easily diffuse, because every cell will require the diffusion of gases. Okay, like plants will also require oxygen for respiration. They will release carbon dioxide. They will also help in the transport of water and minerals, because we all know that conduction of water and minerals and food particles, that is because of xylem and phloem. But lateral conduction, how it is reaching from one cell to another cell, is because of these cells only. Between these cells, like plasmodesmata is there within the within the cell wall and within the spaces, they all are moving. So these cortical cells are helping in the radial conduction also, in the lateral, or in conduction in other different directions also. So so this cortex serves various functions. Now coming to the next layer, which is the endodermis. Now endodermis is also known as the lowermost layer of cortex. Okay, so that all diagram we have drawn before. So in that you have hypodermis, and then you have cortex. Okay, and finally you are coming in the center. So it contains first layer or the outermost layer of the steel, you can say it is endodermis, or you can say it is covering the steel, and it is considered as the lowermost layer of the cortex. So it is made up of parenchymatous cell, okay, but these parenchymatous cells are slightly modified in shape. They are barrel shaped, okay, slightly bigger cells you will find in endodermis. Okay, so they are living cells because they are parenchymatous in nature. They are compactly arranged. Okay, so it's the difference between cortex, that in cortex they were loosely arranged, those parenchymatous cell, but in endodermis they are compactly arranged. The space is not between these cells. Now, yes, this we have talked about that according to some author it is considered to be the innermost layer of cortex. Now, endodermis delimits the steel from the cortex. Okay, definitely it's a barrier between, you can say, between the cortex and the steel. Due to the presence of starch in the endodermal cell, it is known as starch sheath. Okay, now a lot of starch is also deposited in endodermis, and because of it, it is also known as starch sheath. Okay, but yes, there is a difference between monocot and dicot cellular organization and composition. So that all we will discuss when we will talk about those different plant type. The starch sheath is generally, we can see them in dicot plant, in dicot stem. In this endodermis, you will find a very peculiar thing, that is presence of Casparian strip. Now, Casparian strip are, you can say they are strips which is made up of suberin. Okay, suberin is a chemical compound which makes this Casparian strip, you can say ribbon-like structure or strip-like structure, which are deposited on the walls, on the radial and tangential wall of this endodermis. Now, suberin is a lipophilic compound, that is water insoluble. Okay, this figure you can see here that this is a image where you can see, like, this is the cortex, okay, and in the center you have vascular bundles, and this is the endodermis. Okay, in this endodermis you can see thick depositions are there, so they are marking the presence of Casparian strip. So suppose this is an endodermal cell, okay, so these on the radial, suppose on one side I am trying to show it, this Casparian strip is present on the radial and tangential wall of this endodermis. Okay, see, you can see that is the transverse wall and this is the radial wall. So, on these walls, these Casparian strip is present. Now, it is named from on the scientist Caspary, who first noted them in the year 1865. This Casparian strip you can see most prominent in roots. Okay, like, because roots are the point where water is coming inside the root. Water is coming inside the root, okay, through root hairs, okay. So definitely all those cells, this root hair is a single cell, okay, but it is coming in the vascular cylinder, suppose. So this Casparian strip, since it is impermeable to water, so it will help in regulating the flow of water inside the this vascular cylinder. Although it's transferring water, but it's not that every time, even suppose excess water is there, so every time fully loaded with water. So these strips will help in restricting the movement of water. But yes, definitely if everything is bounded around by Casparian strip, so water entry will be completely blocked. So for easy passage, there are some passage cells also wrapped by these strips. So they are allowing the easy entry of water inside the vascular cylinder. So, you will find when you will see the cross section of a root, you will see endodermis as well as facet cell. Now, endodermis will also help in preventing gas bubbles, because suppose if a gas bubble has entered, so because of which has entered into the outermost cells, so it will also be prevented with the help of Casparian strip. It is also important that endodermis will also have a power of cell division. So, that all we will study later, that how this endodermis during time of secondary growth, endodermis or at the time of several other meristematic activity when it is required, this can serve as meristemic stem cells, means those cells which can have the meristematic activity. Now, coming to pericycle, after endodermis you will see pericycle. Generally it is one layer of pericycle is there, and this pericycle surrounds the vascular tissue, and it is considered as the outermost layer of steel. It is also made up of parenchyma, and it is heterogeneous in nature also, because pericycle, see, it might be one layer thick or it might be few layer thick depending on the plant type. Like in dicot stem you will have somewhat multi-layered pericycle, but in roots you will see one layer of pericycle, and it will be made up of parenchyma as well as or sclerenchyma in patches. Sclerenchyma may form discrete bands. Now, pericycle is also very important because it will also have meristematic activity. It's the site of lateral root. So, that time also when we were discussing about root, we have told you that lateral roots, the apical meristem only increases the length of the root. However, the lateral organs were formed from pericycle. So, during secondary growth formation also, this layer will become active and it will give rise to cambium at that point of time. Now, coming to the next important tissue, which in the center you will see that the central portion will be occupied by a parenchymatous tissue which is known as pith. These cells are also isodiametric because parenchymatous in nature, they are thin walled, made up of cellulose, and pith act as a storage or organ also, like storing starch, fatty acid and many tannins also. Then they are, sometimes these may contain fibrates or sclerid, like when a plant gets older, these parenchymatous tissue could be lignified also, and they could be developed into harder tissues. Hollow pith is found in some grasses like in monocot. This pith can degrade, and because of which you can see hollow appearance in case of monocot also. Pith is absent in many dicotyledonous root, okay, and if it is present, it is like that of stem. Then conjunctive tissue is the tissue which is present between, now vascular tissue we will discuss in the next, and but in between the vascular tissue you will see some layers of cells, okay, which are generally parenchymatous and they are known as conjunctive tissue. Then you will have medullary rays. Between these vascular tissues, some elongated cells you will see, these are the medullary rays and they will help in radial conduction. They are also parenchymatous in nature and they connect the pith to the cortex. And generally when your secondary growth formation occurs, you will find a very distinct medullary rays. So, they will be found in wood, and as I told you, they will be involved in transport of water and nutrients. Now, these are the parts of the ground tissue which are found in a plant, but it's not that every type of tissue is present in everything. Like in monocot, it's very important to remember, and that we will discuss also, that in monocot, it's not differentiated into cortex, and means pith, and then medullary rays, pericycle, then you have endodermis. In monocot stem, we will see that the ground tissue is not differentiated in all those parts. You can see just hypodermis, and rest of the tissue is a common ground tissue, and there is no differentiation about pericycle, endodermis, medullary rays and all those things. So, in this lecture, I will just end up here. In the next lecture, we will try to discuss about the vascular tissue which are meant for conduction. So, thank you for today.